islets of Langerhans, alpha, beta, gamma, delta cells, and exocrine functions means, but pancreas release this pancreatic juice, and from this duct it are go with this duodenum, yes, open with this bile duct. But this pancreas has this mixed, the names are mixed glands. But why? You have endocrine and exocrine functions. Also, mixed glands are this male and female sex hormones production, this ovary and testes. Also, I have this both function. Example, ovaries exocrine functions is production this ova, and endocrine functions it produces female sex hormones, example estrogen and progesterone, and males testes are have exocrine function is a spermatogenesis, and your endocrine functions are produced this male sex hormones are this testosterone. Yes, these are two mixed glands. Another are some exocrine glands. Exocrine glands are salivary glands, which are have this duct also, and this duct opening in this mouth and producing the saliva. And most are these endocrine glands, duct-like without duct, and you see how many types of endocrine glands. First time, which is first is a neurotransmitters, which are these chemical messenger systems. Yes, multiply, activate of the cells. Tissue and organs of the body are coordinated by the interplay of several types of chemical messenger systems. Which one? Neurotransmitters. Neurotransmitters are released in end of this axon, in axon terminals of neurons, into the synaptic cleft. And the example is neurotransmitters, acetylcholine, neurotransmitters, norepinephrine, many neurotransmitters which are released from axon terminal into the synaptic cleft. And this next is a presynaptic membrane, and in postsynaptic membrane are locations this the same this receptor with this neurotransmitter. Example, if acetylcholine neurotransmitters, then receptors on nicotinic and muscarinic receptors. If norepinephrine receptors is alpha and beta adrenoreceptors, which are also have your subtypes alpha 1, alpha 2, beta 1, beta 2, and other. Neuromuscular junctions, example, when this neuron are connected with this muscle. Yes, it sends impulse from your action potentials and release this neurotransmitter acetylcholine, example. Next is endocrine hormones are released by glands or specialized cells into the circulating blood to influence the function of target cells and at another locations in body. But target cells not all times are location near this endocrine glands. Example, anterior pituitary glands, which are producing this.
Hello, doctor. Good
Yes.
morning. Yes, sorry,
morning.
Sorry, doctor, to stop you there, to
Yeah,
interrupt you a bit.
no problem.
Yeah, we're just asking a question. Is this going to be part of the midterm 2 exam as well?
Which repeat please questions.
Is this part of histology going to be part of midterm 2?
If endocrine systems.
Yes, is this part of midterm 2?
No, in midterm 2, yes, part of endocrine, yes. Urinary and endocrine systems, two systems, urinary and endocrine. Yes.
Thank you very much. Okay.
Endocrine is very interesting systems, I think. I love very these systems. It has this many hormones. You study this all gland functions and actions, different types of chemical neurotransmitters. Use this in this midterm exams. You found in your guide and review books 2 chapters, which is this urinary systems and endocrine systems. Both systems, two systems, MCQ. This in this book are also, has this explanation. I small change these questions, prepare these all questions, and you all students are passed these exams, no problem. Okay, endocrine hormones, I tell, which are released by glands and specialized cells into the circulating blood, and these neuroendocrine hormones. What means neuroendocrine hormones? Are secreted by neurons into the circulating blood and influence the function of target cells at another location in the body. Okay, these are third types. Fourth, paracrines secreted by cells into the extracellular fluid and affecting neighboring target cells of a different types. Autocrines means secreted by cells into the extracellular fluid and affecting the function of the same cells. And cytokines, with peptides secreted by cells into the extracellular fluid and functioning as autocrines, paracrines, and endocrine hormones. Example of cytokines include these interleukins. Interleukins, which are help these T lymphocytes activate from B lymphocytes. Interleukins then make this T lymphocyte clone and other actions. Repeat this all endocrine glands. But first time tell, but endocrine glands are have this your receptors and two types: primary endocrine organs and secondary. What means primary? Whose primary function is the secretion of hormones. These are named endocrine glands. And secondary endocrine organs, which the secretion of hormones occurs secondary to some other functions. Example, primary endocrine organs, hypothalamus, 1, are located within the brain, including pituitary, hypothalamus, pineal, thyroid glands, parathyroid, thymus, adrenal glands, pancreas, and gonads, testes, and ovaries. Secondary endocrine glands include organs, heart, liver, stomach, small intestine, kidney, and skin. I tell about all hormones which are produced by these organs. Some of these secondary hormones which are produced by organs which are primary functions is not are secretion of hormones. Yes, example, heart. Heart's primary function is pumped blood into and out this in body. Yes, in this from aorta in this whole body and from this pulmonary trunk to this lungs. Yes, and then all body are oxygenated blood carries and deoxygenated from superior inferior vena cava are received into the right atrium. These are primary function. Yes, pumped blood, and this also atrium are producing atrial natriuretic peptide, and these are function are decreased blood pressure, opposite function of aldosterone. These are names secondary hormones. Yes, primary and secondary. Hypothalamus, 1. Hypothalamus is a example of this neuroendocrine regulation. Why? You have the cells in hypothalamus, yes, which are producing these hormones which are help regulate another hormones. Example, hypothalamic hormones, lateral hypothalamus has the cells which are producing releasing hormones, which are activate action of anterior pituitary gland, and also has this inhibiting hormones, which are also inhibits releasing from anterior pituitary glands. Example, these are hypothalamic releasing hormones. Which one? Growth releasing hormones. Growth releasing hormones. GRH. Yes, which are help release from anterior pituitary glands growth hormones. 1. Second, corticotropin releasing hormones, CRH, activate anterior pituitary glands, which is names adenocorticotropic hormones, ACTH, anterior pituitary glands. These are hypothalamus. Two, GRH, CRH. Next, TRH, thyrotropin releasing hormones. I explain about hypothalamic hormones, which are produced from these cells of hypothalamus. Yes. And TRH, thyrotropin releasing hormones, activate TSH, thyrotropin stimulating hormone from anterior pituitary glands. Three. Fourth, gonadotropin releasing hormones, GnRH. Gonadotropin releasing hormones function activate anterior pituitary glands. Gonadotropic hormones, which are two types, is follicle stimulated hormones, FSH, and luteinizing hormones, anterior pituitary gland hormones. These four releasing hormones, which are activate action of anterior pituitary gland. Also has these two inhibiting hormones. One inhibiting hormones is the names growth inhibiting hormones, GIH, which are also named somatostatin, and second, prolactin inhibiting hormones, PIH, which are also are named dopamine. These are, and sometimes seven hormones are tells, but is a hormones which are activate anterior pituitary gland hormones, prolactin. Okay, prolactin inhibiting hormones and prolactin releasing hormones. And these are all hormones which are produced from hypothalamic cells, and it activate anterior pituitary gland hormones. Next is a pituitary glands. Pituitary glands or hypophysis names, which are has these two lobes, anterior lobes and posterior lobes. About anterior lobes hormones, I repeat these all hormones, which is GH, growth hormones, ACTH, adenocorticotropic hormones, GnRH, no, sorry, not GnRH, is a R means releasing, is a hypothalamic. These are gonadotropic hormones, gonadotropic hormones, which are two types, is a luteinizing hormones and follicle stimulated hormones. Next, TSH, thyrotropin stimulated hormones, prolactin, and prolactin, and one is a melanocyte stimulating hormones, which are sometimes are tell, but is intermediate parts of the pituitary gland, but in most authors are tell, but is anterior pituitary glands, which are help are regulation of melanocytes in the skin. Okay, understand? Yes. Pituitary glands hormones, growth hormones. Next, ACTH, adenocorticotropic hormones, gonadotropic hormones, which are two types, is FSH and LH, prolactin, ACTH, adenocorticotropic hormones, and last is a melanocyte stimulating hormones, and TSH, thyrotropin stimulated hormones. These are pituitary glands anterior. What is the posterior lobes? Posterior lobes, remember, it's not producing these hormones. These two hormones are producing by also hypothalamic, supraoptic and paraventricular cells. What is his name? ADH, antidiuretic hormones or vasopressin, which are after from axon transports, and this oxytocin from paraventricle and supraoptic and paraventricular cells are produced two hormones, oxytocin and vasopressin. These both hormones are traveled down from axon transport in the posterior lobe. But posterior lobe of pituitary gland did not have hormone producing cells. It's a axons and axon terminal. But cell body of these neurons are located in hypothalamus, and hypothalamus and pituitary glands are connected with this infundibulum. Yes, and from axon transports travel down these hormones and storage in this axon terminal, and after release in this blood. It's only function of release functions, storage in axon terminal and release in this blood. Remember, posterior pituitary glands did not producing hormones, only release. Its two hormones are production in hypothalamic cells, which are named supraoptic and paraventricle cells. In total, in hypothalamic cells, lateral hypothalamus and these supraoptic and paraventricle cells are producing how many? Two, oxytocin and antidiuretic hormones or vasopressin, and the seven hormones, which are 5 releasing hormones and 2 inhibiting hormones. Yes, 7 plus 2, total is 9 hormones are producing from hypothalamic cells. Releasing hormones and inhibit some activate this anterior pituitary glands hormones. And inhibiting hormones also activate anterior pituitary glands hormones. Inhibiting hormones in hypothalamus too. Which one? GIH, growth inhibiting hormones, or somatostatin, and PIA, prolactin inhibiting hormones, which are also named dopamine. Okay, understand? Pineal glands are producing only one hormone. It's melatonin. Remember this, which are help sleeping and another activity. Thyroid glands are producing 3 hormones: T3, tetraiodthyronin, 3-iodthyronin, T3, T4, tetraiodthyronin or thyroxine, 3-iodthyronin, T3, and calcitonin, which are regulate calcium levels. No, it's more regulate is parathyroid glands, but parathyroid glands are 4 small glands in posterior parts of this thyroid gland, which are producing only one hormones, parad hormones. First time, remember, and I think you know this names, all hormones which are producing from this endocrine glands. Yes, hypothalamic hormones, pituitary glands, anterior, posterior, pineal glands, 1 hormones, thyroid glands, 3 hormones, T3, T4, and calcitonin. Parathyroid glands, 1 hormones, parad hormones. Next, thymus, also thymosin sometimes, which are helps regulate, differentiate this T lymphocytes. Next, adrenal glands, which are has this outer cortex and inner medullary parts. Cortical hormones, cortex are have these 3 zones. These are zona glomerulosa, zona fasciculata, zona reticulare. Zona glomerulosa producing mineralocorticoids and one of this aldosterone, which are help reabsorption of sodium ions from these tubules to the blood. These peritubular capillaries, and also reabsorption of water. Next is glucocorticoids, which zona fasciculata, which are produce glucocorticoids, and one of these glucocorticoids is a cortisol, corticosterone. And third zone is a zona reticularis, which are producing this sex androgens and small concentration estrogen also. Next, about pancreas, which are these mixed glands. I tell this, which are have this both function, exoendocrine functions, with duct and without duct. Endocrine functions, 4 types of cells, isolates of Langerhans. Also are sometimes some students are tell Langerhans cells. Langerhans cells also are in the skin, which are this has this microphage of this skin. Yes, it's Langerhans cells. and the islets of Langerhans, which has alpha, beta, gamma, delta cells, produce 4 types of hormones. Which one? Alpha, glucagon, beta, insulin, delta, somatostatin, and gamma is pancreatic polypeptides. Next is ovary. About ovary, I tell this producing 2 hormones. These are estrogen and progesterone, and uterus also are producing some of the hormones, and testes are producing this testosterone. These are all endocrine glands with your hormones. Please remember all hormones name, okay? Endocrine glands are like the duct, secrete the product into the blood, produce biologically active molecules called hormones. The blood carries the hormones to target cells, and this contains specific receptor proteins for the hormones, or has this action with this. Some hormones need this secondary messengers. Yes, primary is hormones, and secondary messenger sometimes is a cycling AMP, calcium ions, calmodulin. Next is a tyrosine kinase, and other. I explain late about action of different types of hormones, which are have these different chemical structures. But these important hormones are bind with these receptor proteins. Okay, some need second messenger systems, some need some without second messenger systems activate this G protein systems, and which are sometimes open and close these ion channels. About primary endocrine and secondary endocrine organs, I tell these are hypothalamus, pituitary, pineal, thyroid, parathyroid, thymus, adrenal, pancreas, and gonad, testes in the male and ovaries in the female. Remember, but 3 zones which are regulate this release hormones. First, hypothalamus example, yes, which are producing gonadotropin releasing hormones. Releasing hormones activate anterior pituitary gland gonads, FSH and LH. And this FSH and LH then activate this gonads of this male and female sex hormones. Testes in the male and ovaries in the female. You see these 3 zones: hypothalamus, pituitary glands, and this target tissue with your target cells, with your specific receptors for these hormones. Secondary endocrine organs is the heart, liver, stomach, small intestine, kidney, and skin. Which hormones are produced? This all. No, these are about this all glands, and you see. Next, I very interesting this table about these all hormones, which you know, which I tell this primary and secondary. You know, adipose tissue also are produced these major hormones, which are named leptin. And target organs is hypothalamus. And what is it? It suppresses appetites. But you know, in hypothalamus has this eating center and satiety centers. Yes, when activate eating centers are obesity of the people, but it's eating more. And if these satiety centers are activate, yes, then is names aphasia. Aphasia is not eating. Yes, it's and sometimes as this leptin, which are produced adipose tissue, are these are suppresses appetite. Okay, sorry. Next, about adrenal cortex, I tell these 3 types of hormones. These are glucocorticoids and aldosterone, mineralocorticoids, aldosterone, and also sex hormones, males androgen, adrenal medulla, epinephrine and norepinephrine, heart with this secondary endocrine organs, which are producing this atrial natriuretic peptide hormones. Yes. Next, hypothalamus releasing and inhibiting hormones. I explained this about all hormones. Next, a small intestine, GI tract hormones, which are help digestion or producing help this production these enzymes. Example: secretin, cholecystokinin. These are all gastrin in the stomach. These are in small intestine are producing the secretin and this in stomach gastrin. This one in testis testosterone. Thymopoietin also are in this thymus, thyroid glands, T3, T4, and calcitonin, pituitary, posterior, two hormones only storage, not producing and release these two hormones, these anti-diuretic hormones, vasopressin and oxytocin, which are producing in this hypothalamic, supraoptic, and paraventricle cells. Next is a pituitary anterior glands with eutrophic hormones. I tell about all hormones. Pineal glands, one, melatonins. Next is a parathyroid glands. Is parathyroid hormones. Ovaries. This estradiol, estrogen, estriol, many types, and progesterone. In liver are also one hormones are produced, which are named somatomedins. Yes, somatomedins, which are this primary target tissue organs is in the cartilage, and which are stimulation cell division and growth in this liver. In kidney, are producing this erythropoietin. Erythropoietin is a hormones which are help which where is location primary target organs in the bone marrow. It helps this erythropoiesis. Erythropoiesis are formation of RBC. Next, isolates of Langerhans cells, I tell insulin and glucagon alpha and beta cells, beta insulin alpha glucagon, also delta and gamma cells. In small intestine, I tell secretin and the cholecystokinin, hypothalamic cells, I tell in heart natural. Okay, understand? These all are hormones which are in primary and secondary organs are produced. Now, these are some hormones, specialized neurons, particularly in the hypothalamus, secretes chemical messengers into the blood, raises them into the neurosynaptic cleft. In these cases, the chemical that the neurons secrete is sometimes called neurohormones. Neurons are released neurohormones. In addition, a number of chemicals, for example, norepinephrine, are secreted both as a neurotransmitter and as hormones. Neurotransmitters with this axon terminal and also neurotransmitters which are released in postganglionic sympathetic neurons, also norepinephrine, and adrenal medulla. In adrenal glands medulla, which have released this epinephrine and norepinephrine, and these are certain types of action also. Neuroendocrine cells, its name. Hormones affect the metabolism of their target organs and by the means help regulate total body metabolic growth and reproduction.
Sorry,
Yes.
Doctor. Sorry, is this slides on Drive already? Do we have this access to the slides?
Slides.
Yeah,
Yes.
I mean this presentation, is it on Drive, or
Yes.
are you going to send it to us?
Yes, I have this our presentation, and in the next hour today's lecture, I open this interactive physiology, and you show the slides also. Okay. Yes, you understand about slides. You tell me or what?
Yeah.
Yes.
Yes, about slide. Yes, we're just
Okay.
thinking maybe it's on the Drive, whether we can have access to it already.
Okay, wait, and you also see the slides. Next is this chemical categories of hormones, amines, polypeptides, glycoproteins, and steroids. No, these are classification of chemical. These hormones, which are amines, there are hormones derived from amino acid tyrosine, and tryptophan. You know, our his amino acids are 20 times. Yes, one of this tyrosine and tryptophan. And these hormones, which have these amines, amino acids, are tyrosine, amino acid tyrosine, and tryptophan. Polypeptides and proteins. Peptides, you know, peptides is a some of this bind some of amino acids. Yes, peptide, tripeptide, dipeptide, oligopeptide. 100 amino acids is named proteins. Yes, polypeptides. When these peptides are bind each of other proteins are large polypeptides. Large polypeptides, example, 100 amino acids. Yes. So this distinction between the two categories is some what are example arbitrary. If a polypeptide chain is a target about 100 amino acids, it's called protein. Next, glycoproteins. These molecules consist of protein binds to one of more carbohydrate groups. And you see these mixed words, glycoproteins, proteins and carbohydrates, glycogen example. And for steroid hormones are derived from cholesterol after an enzyme cleaves off or cuts the side chain attached to the 5-carbon dearranged. It's named steroid hormones. No, these are two types also water soluble or polar, the hormones that are polar and therefore water soluble hormones, and the hormones that are non-polar and this insoluble in water, because the non-polar hormones are soluble in lipids and they are often referred to lipophilic hormones. Some hormones which are names non-polar, insoluble hormones in water but soluble in these lipids. It's named lipophilic hormones. Philia means love, yes, phobia, opposite. And these polar, therefore, are water soluble molecules. Two types, water soluble and these are classification of hormones. Next, lipophilic hormones include the steroid hormones and thyroid hormones. Steroid hormones and thyroid hormones. Steroid hormones is adrenal glands cortex hormones. Steroid hormones are secreted by only two endocrine glands, is adrenal cortex and the gonads. Gonads, these male and female sex hormones, and cortical hormones are three types. Yes, mineral corticoids, glucocorticoids, and this and androgens. And thyroid hormones are secreted by thyroid glands. Steroid hormone, chemical lipophilic hormones are steroid hormones and thyroid. Thyroid hormones released from thyroid glands, T3, T4. And the steroid hormones are adrenal glands and gonads. Steroids and thyroid hormones are active when taken orally as a pill. By contrast, polypeptides and glycoprotein hormones cannot be taken orally because they would be digested into inactive fragments before being absorbed into the blood. Polar or water-soluble hormones include polypeptides, glycoproteins, and the catecholamine hormones secreted by adrenal medulla. Catecholamines named epinephrine and norepinephrine. And water-soluble hormones include polypeptides and glycoproteins and catecholamines. This all. These hormones are too polar to pass through the phospholipid portion of the plasma. You know this structure of plasma membrane. Yes, this bilayer of phospholipids, and bind the receptor on the surface of the cells. These are precursor molecules. These names prohormones. And for example, insulin is produced from proinsulin within the beta cells of the islets of Langerhans on the pancreas, prohormones, and then pre-pro-insulin. Example, in some cases, the prohormone itself is derived from an even larger precursor molecules. In the case of insulin, the molecule is called pre-pro-insulin. to term pre-hormone is sometimes used to indicate such precursors of prohormones, pre-prohormones, then prohormones, and last hormones. These three three gradation: pre-prohormones, next inactive forms also prohormones, and last is the hormones. Precursor means parent. Prohormones is precursor of a hormone, and pre-hormone are precursor of prohormones. Prohormones, prohormones, and then last hormones. Now about target cells have mass specific receptor proteins that combine with the regulatory molecules. And combination of the regulatory molecules with its receptor protein must cause a specific sequence of change in these target cells, and the must be the mechanism of action of the regulator. A mechanism which involves rapid removal and or chemical inactivation of the regulator molecules is essential, because without off-switch physiological control would. Now this hormone interaction, next, three types of actions. Interactions include synergistic, when two or more hormones work together to produce a particular result. Permissive, when one hormone increases the activity of the second hormone. And antagonistic effects. Synergistic effects, permissive effects, and antagonistic effects. Example, when two or more hormones work together to produce a particular result, effect is named synergistic. This synergistic effect may be additive of complementary action, or epinephrine and norepinephrine of the heart is a good example of additive effects. Next, estrogen, ability of the mammary glands to produce and secrete milk in lactation requires the synergistic action of many hormones: estrogen, cortisol. Estrogen is a female sex hormone which are produced from this ovary. Cortisol is the adrenal cortical hormones which are named this glucocorticoids in the zona fasciculata. Prolactin. Prolactin is a anterior pituitary gland hormones, and oxytocin. Oxytocin, which are produced in hypothalamus and its storage in this posterior lobe of pituitary gland, which have complementary actions. Estrogen, cortisol, prolactin, and oxytocin. That is, each of these hormones promotes a different aspect of mammary gland function, so that their cooperative effects are required for lactation. A hormone is said to have a permissive effect on the action of a second hormone when it enhances the responsiveness of a target organ to the second hormones, or when it increases the activity of the second hormones. Prior exposure of the uterus to estradiol, major estrogen, sometimes is precursor of estrogen, includes the formation of receptor proteins for progesterone, which improves the response of the uterus when it is subsequently exposed to progesterone. This estradiol has a permissive effect of the responsiveness to the uterus to progesterone. This example, vitamin D3 is a pre-hormone that must be modified by enzymes in the kidneys and liver, where two hydroxyl groups are added to form the active hormones, 1.25 dihydroxy vitamin D3. This hormone helps to raise blood calcium levels. Parathyroid hormones, which are parathyroid gland hormones, has a permissive effect of the action of vitamin D3 because it stimulates the production of hydroxy-linked enzymes in the kidney and liver. By this means, an increased secretion of parathyroid hormones has a permissive effect to ability of vitamin D3 to stimulate the intestinal absorption of calcium. No antagonist affects insulin and glucagon. You know these two hormones of the pancreas. Alpha cells glucagon are produced, and beta are insulin. Two hormones from the pancreas is isolates of on adipose tissue. The formation of fat is promoted by insulin, whereas glucagon promotes fat breakdown. The high concentration of estrogen in the blood inhibits the secretion and action of prolactin. So, lactation during pregnancy is inhibited. Some hormones like norepinephrine and epinephrine are secreted within seconds after the gland is stimulated and may develop full action within another few seconds to minutes. On the other hand, the action of other hormones such as thyroxine or tetraiodthyronine or growth hormones, anterior pituitary gland, these are thyroid gland hormones, may require months for full effect. But action of this also neurotransmitters, which are have this effects after few seconds and sometimes of the months are have difference. No half time of a hormone, the time required for the plasma concentration of a given amount of the hormone to be reduced by half range from minutes to hour. For most hormones, thyroid, however, has a half time life of several days, and some are several times. Hormones are present within their normal or physiological range of concentrations when some hormones are taken in abnormally high or pharmacological concentration, as when they are taken as drugs. The effects may be different from these products by lower, more physiological concentration. You know, these are concentration in the blood are range from as little as 1 picogram, 1 million of a grams in each milliliter of blood, up to at most a few micrograms, a few millions of a gram per milliliter of blood. Similarly, the rates of secretion of various hormones are extremely small, usually measured in micrograms and milligrams. Yes, well, some effects are number of these receptor proteins has this upground regulation and downground regulation. When increases number of receptors which are bind its hormones, its upground regulation names, and when decreases the concentration of hormones, then decrease it activates this more iron receptors. And the downground means are when high concentration levels of hormones in blood, then the receptors, number of receptors are decreased. It's named downground regulation. As this I tell, this is priming effect causing in large part by the upregulation of receptors. In priming process, increased number of receptor proteins for the hormone being primed are inserted into the plasma membrane. And down regulation when this desensitization is partly towards the fact that high concentration of these hormones cause a decreased number of receptors is named down regulation, up regulation, and down regulation. Mechanism of hormone actions I explain after 10 minutes, okay? A 10 minutes break time, and after I continue explanation of action of this different type of neurotransmitter. It's very interesting. Okay, 10 minutes break time. Yes.
Yes, doctor. 10 minutes fine. We'll come back after 10 minutes.
After 10 minutes.
Thank you.
Okay.
Thank you, doctor.
Hormones influence target cells by binding to specific receptors. The hormone-receptor interaction initiates the process of transforming the chemical message into the response of the cell.
These receptors, which target cells, are located in the different space. One, you see located in this plasma membrane. Second, inside the cytoplasm. And third, you see inside this nucleus. These are DNA, near this DNA, three types.
A target cell responds to a hormone with a sequence of events. Drag the hormone to the receptor to observe the events and response of the target cell. Receptor activation by the input signal, the hormone. Conversion of the input signal to a biochemical change in the cell. Conversions occur through second messengers and gene expression. Response of the cell. Examples of cellular response include contraction, secretion, transport, and synthesis or breakdown of storage molecules.
Yeah.
We will study receptors here and conversion mechanisms and cellular responses later in this topic. Click Continue to proceed.
No, first time these receptors, which are located in the different types of cells, plasma membrane, inside cytoplasm, and inside this nucleus. When hormone binds these receptors, then have these different types of response after. No, different types of actions. First time you see these G protein systems, and when GDP is converted with GTP bind, GTP has this high energy. It activates adenylate cycles. Adenylate cycles are convert ATP with ADP. Then ADP with cycling AMP. Cycling AMP is the second messenger. Second messenger activate protein kinase inside the cytoplasm, and protein kinase are activate this phosphorylation of these proteins, different types of action. But also our response, how many types? Contraction of blood vessels, secretion, some of these neurotransmitters, transport, you see this active transport, and synthesis, these proteins, these are amino acids, yes, and sometimes are breakdown.
Receptors are complex proteins, each specific for a particular hormone that binds with a high affinity. Hormones only affect target cells, cells that have specific receptors for them. For ease of viewing, receptors and hormones are drawn as simple shapes. Click the enlarged receptor to continue.
These oral receptors are located in this.
Receptors are located in the plasma membrane or in the interior of the cell. Water-soluble hormones cannot diffuse into cells. They bind to receptors in the plasma membrane. Drag the water-soluble hormones to their receptors.
No example water-soluble is catecholamine, example epinephrine and norepinephrine, and also insulin, which are also are water-soluble. Okay, sorry, one.
Lipid-soluble hormones diffuse into cells where they bind with intracellular receptors. Drag the lipid-soluble hormones to their receptors.
These are cortisol and thyroid. These are lipid solubles. One in thyroid. These are inside this.
Recent evidence indicates that there are also receptors in the plasma membrane for lipid-soluble hormones and that these receptors may induce more rapid responses of the target cell than intracellular receptors. Target cells have receptors for multiple hormones, and each hormone exerts a different action on the cell. Also, a given hormone may act on the same cell through different receptors, each inducing a different action of the cell. Not all cells possess receptors for all hormones. The number of receptors present in a cell may vary depending on the amount of hormone present. Prolonged high hormone levels may decrease the number of receptors and reduce the response in target cells, a process called downregulation. Continuous low levels of a hormone or another stimulating signal may increase the number of receptors and the sensitivity of the target cell, a process called upregulation. For example, hypersecretion of thyroid hormone upregulates receptors for epinephrine. This phenomenon leads to the increased heart rate and cardiac output seen in patients with hyperthyroidism. Click a thyroid hormone to see how hypersecretion upregulates receptors for epinephrine. Receptors are dynamic proteins, and like many other cellular proteins, they are recycled and replaced repeatedly during the life of a cell.
So you see this action of upregulate and downregulate means. Upregulate means when increases number of receptors which are bind these hormones, and the downregulate means when decrease the number of receptors which are located in these different types of space of the cells.
The water-soluble hormones, peptides, and catecholamines.
1 minute. You see this first time, this action of water-soluble molecules. These are hormones, and your target tissue cells, and in tissue are cells, these receptors. When these hormones are bind with this receptor, then you see inside this plasma membrane, you see this it's a change shape, and deletes this GDP and binds this GTP, guanosine triphosphate. These are guanosine diphosphate. After guanosine diphosphate activates these enzymes which are named adenylate cyclase. This one, adenylate cyclase are convert this ATP with cycling AMP. Cycling AMP or CAMP, adenosine monophosphate, is a second messenger. The second messenger activate in cytoplasm are second enzymes which are named protein kinase. Protein kinase after are activated phosphorylations is another proteins. See please this animation, and then I again explain this all events.
Plasma membrane receptors on the outside surface of the cell. There are several categories of plasma membrane receptors, including receptors that activate G proteins and receptors with protein kinase activity. We will examine the most common G protein mechanisms and the protein kinases called tyrosine kinases that phosphorylate tyrosine. We will look at each kind of receptor, beginning with the receptors that activate G proteins. In the inactivated state, guanosine diphosphate, GDP, is bound to the G protein. Drag the hormone to the binding site on the receptor to observe its action.
Drug hormones with this receptor.
When a hormone binds to the receptor changes shape and activates the G protein. It releases GDP and binds guanosine triphosphate, GTP, a high-energy molecule. Part of the activated G protein travels along the membrane and activates an enzyme called adenylate cyclase. Adenylate cyclase catalyzes the conversion of adenosine triphosphate, ATP, to cyclic adenosine monophosphate. Cyclic AMP is a second messenger. The hormone is the first messenger. Cyclic AMP activates the intracellular enzyme protein kinase A, which can phosphorylate many other proteins, activating some and inhibiting others. This mechanism can induce many different effects within a single cell and different actions in different cell types. This second messenger system can also phosphorylate ion channels and alter their activity. The response of the cell ends when cyclic AMP is degraded to AMP in a reaction catalyzed by the enzyme phosphodiesterase. While active, G proteins keep stoking the pathway to phosphorylation of cellular proteins so that huge numbers of them are produced. This phenomenon, called amplification, greatly increases the effect that a single molecule of a hormone can have on a cell. To view the animation again, drag the hormone to the receptor. Click Continue to proceed.
Repeat this whole process. First time when these hormones bind to this receptor, activates these G proteins. G proteins then are convert GDP with this GTP, guanosine triphosphate. This activates an plasma membrane enzyme adenylate cyclase. Adenylate cyclase are convert this ATP with this cycling AMP. Cycling AMP then activate second enzymes, which are named. 1 minute, I drag and this animation GDP, adenylate cyclase, convert ATP with cycling. Cycling AMP activates protein kinase. Protein kinase are phosphorylate these proteins, and also sometimes it inhibits these some channels. And the end of this process, when these phosphodiesterase enzymes are convert cycling AMP with adenosine monophosphate, and these are guanosine diphosphate also bind with this G proteins. Okay, repeat this whole process. Repeat or understand. Repeat this all events when this hormone are bind with this receptor. Please answer yes or no.
No need, we understand.
Understand. You understand. And other students also understand? Only one student listening me? Okay. Now, if you understand, I continue the second types of action.
G proteins, like the one we just described, that stimulate production of cyclic AMP are known as stimulatory G proteins. Inhibitory G proteins inhibit production of cyclic AMP and the subsequent response of the cell. Another type of G protein activates an intracellular messenger system that uses diacylglycerol and inositol trisphosphate as second messengers. Drag the hormone to the binding site on the receptor to observe its activity.
Second messengers are second type of action are sometimes calcium ions, calmodulin, and in this case, another G proteins activate an intracellular messenger systems which are that uses diacylglycerol (DAG) and anisotol triphosphate (IP3) with the second messengers. When drugged this hormone,
when a hormone binds to the receptor changes shape and activates the G protein. It releases GDP and binds GTP, a high-energy molecule. Part of the activated G protein travels along the membrane and activates an enzyme called phospholipase C. Phospholipase C causes a membrane phospholipid called phosphatidylinositol bisphosphate to cleave into diacylglycerol and inositol trisphosphate. Each functions as a second messenger. Diacylglycerol remains in the membrane where it activates protein kinase C and inositol trisphosphate enters the cytosol. The activated protein kinase C phosphorylates other proteins, causing responses of the cell. Inositol trisphosphate prompts the endoplasmic reticulum to release calcium ions. Calcium ions also behave like second messengers. They can act directly on proteins or bind to the protein calmodulin that can act on proteins to cause the response of the cell. To view the animation again, drag the hormone to the receptor. Click Continue to proceed.
The first time explains this whole process. Hormone bind with this receptor, then change shape inside this plasma membrane, activate G proteins. G proteins release GDP and binds GTP. GTP is then activates this phospholipase C, and phospholipase C, which are converted with these two types of enzyme, diacylglycerol glycerol, and the second is IP3, anisotol triphosphate. Diacylglycerol activates this protein kinase, which are phosphorylate as other enzymes, and IPT are go inside the cytoplasm with this endoplasmic reticulum and release calcium ions. And calcium ions also are believes is like a second messenger calcium ions, and it can directly on proteins. It's in proteins named calmodulin. It binds with this another second messenger also calmodulin, and that act of on proteins to cause the response of the cells. Okay, as this whole process, I bind these hormones with these receptors and see this animation of all process. Anisotol triphosphate, diglycerol, which are activate protein kinase, it's responsible of the cells, calcium release of endoplasmic reticulum bind with this calmodulin, which are also, and this another old process. Okay, understand this action of different types of hormones which are has this say in second messenger calmodulin and calcium ions, calcium calmodulin ion second messenger. In this first time which I explained, the second messenger is cycling AMP, which also regulates this protein kinase. In here, phospholipase C, which are activate these two enzymes, DIG and IP3. DIG activate this protein kinase C in this plasma membrane also there, and IP3 anisotol triphosphate enter in this cytoplasm, activate this endoplasmic reticulum, release calcium ions. Calcium is a second messenger which are the response of these different types of action inside the cells, and also it's bind with this calmodulin, and calcium calmodulin also has this different action. Okay, this please again show this animation.
Drag each hormone to a binding site on the receptor to observe its activity.
Activity of this insulin molecules, which are need this.
When a hormone binds to a tyrosine kinase receptor, the receptor joins with a neighboring receptor and changes shape. Each receptor phosphorylates tyrosine groups in its neighbor. The process is called autophosphorylation because the receptors are of the same type. The phosphorylated tyrosine groups provide docking sites where relay proteins bind. They initiate a series of protein phosphorylations that lead to the response of the cell. Receptors with tyrosine kinase activity often bind hormones like insulin and other growth factors that affect cell growth and differentiation. To view the animation again, drag the hormone to the receptor. Click Continue to proceed.
You drag, then it join each of other and change shape, then phosphorylation, these receptors, and phosphorylation are needs this and other proteins, which are, 1 minute, which is continue.
On this page, we will study mechanisms used by epinephrine and insulin.
Okay. I also show this mechanism when open your books, one of these books, Irafox, and repeat again this action. Very interesting slides are have in your books. This example one and this one. When these hormones bind to this receptor protein, it activates G protein systems, G proteins which are released GDP and GTP. GTP are hydrolysis G proteins, 3 subunits from alpha and beta gamma subunits. Alpha subunits activate adenylate cyclase. Adenylate cyclase then ATP converts with the cycling AMP and release inorganic phosphate. After cycling AMP, which are second messenger, first messenger, second messenger, are regulate subunits which are activate protein kinase inside the cytoplasm, and it's regulatory subunits of cycling AMP, and sometimes it's protein kinase if in activation, and sometimes activate, which are phosphorylation of proteins, activation of specific enzymes, or inactivation of specific enzymes. These are action of one of these neurotransmitters. When activate, when second messenger is cycling AMP, which are converted from this adenylate cyclase neurotransmitters, enzymes, sorry, which are located in this plasma membrane. Second types you see when second messenger is calcium calmodulin complex. First hormone binds to these receptors in the plasma membrane of the target cells, these target cells and these receptors. Next, dissociation of G proteins, which are activate phospholipase C, which are location in this plasma membrane, yes? And the phospholipase C protein subunit travels through the plasma membrane and activates phospholipase C, which catalyze breakdown of a particular membrane phospholipids into dicyl glycerol or DIG and IP3, anisotol triphosphate. Then anisotol triphosphate, this one, enters into the cytoplasm and bind to receptors in the endoplasmic reticulum, causing release these calcium ions. And calcium ions then diffuse into the cytoplasm, where it acts as second messenger to promote the hormonal effects in the target cells, and also is bind with this calmodulin and calcium calmodulin complex are have this another different type of actions. Okay, these are second types when activates phospholipase C, which are catalyzed reactions. Particular membrane phospholipid into dicyl glycerol and IP3. These are second action of neurotransmitters. After also this norepinephrine and epinephrine or adrenergic receptors action, which has this two types of receptors, and action are two different types. First, when beta adrenergic effects of epinephrine, when epinephrine are bind with this beta adrenoreceptors, but you know receptors from this norepinephrine and epinephrine are two types, alpha and beta, with your subtypes. When this norepinephrine binds with this beta receptors, then adenylate cyclase is activate, converts ATP with cycling AMP. Cycling AMP active protein kinase. This one is same with this when alpha adrenergic effects of epinephrine bind. Yes, then the second types you see this IP3 and anisotol triphosphate when releases calcium ions, IP3 from endoplasmic reticulum when are storage this calcium ions, and calcium ion bind with calmodulin. Calcium calmodulin activate also protein kinase, and both actions which are active protein kinase and this active protein kinase from beta receptors and from alpha receptors are then bind with this glycogen and convert this glycogen from glucose 1 phosphate, which are converted with glucose 6 phosphate, and then are release this free glucose, and it go in this outside these cells. This all are binding of epinephrine to beta adrenergic receptors, activates adenylate cyclase, and leads to the production of cycling AMP, which activates a protein kinase, and protein kinase binding of epinephrine of alpha adrenergic receptors leads to a rise in cytoplasm calcium concentration, which activates calmodulin. Calmodulin then activates this one protein kinase also, which like the protein kinase activated by cycling AMP. These are two from cycling AMP and from calmodulin. Alters enzyme activity so that glycogen to convert it to glucose 6 phosphate, and the glucose 6 phosphate group is removed by another enzymes, so that the liver cells secrete this free glucose into the blood in response to epinephrine. Okay, epinephrine sometimes are increases glucose levels. And tyrosine kinase also last, which are explain action. You see this receptor of insulin. You need these two molecules of insulin. Insulin receptors consist of two parts, each containing a beta polypeptide chain. You see this alpha polypeptide chain, and out beta and alpha. Alpha this one and beta this one. Alpha binds these insulin molecules, and beta polypeptide chain that spans the membrane, and alpha chain that contains the insulin binding sites. When two insulin molecules, sizes 1 and 2, bind to the receptor, the two parts of the receptor are phosphorylate each of other. Yes, you see this. And this greatly increases tyrosine kinase activity. Tyrosine kinase active phosphorylation of receptor, second steps, and third step, phosphorylation of signal molecules are cascade of effects, and one is glucose uptake and anabolic reactions, and the signal molecules that produce a cascade effects. Okay, these are protein kinase, and also one of these mechanisms. These are thyroid hormones. Okay, 1 minute. Okay. About this IP3 and diacylglycerosis cycling AMP, I explained. And last, this one, mechanism of thyroid hormones. Thyroid hormones, you know, are two types. Yes, these are T3 and T4. When these thyroid hormones are entering the blood, these are blood and these are cells. Yes, target cells and this blood. In blood is bind with this thyroglobulin, and the T4 first thyroxine are carried to the target cells, bind to its plasma carrier proteins. These are one of these are globulin, immunoglobulin. After is dissociation from its carrier, and they pass through the plasma membrane, entering plasma membrane of its target cells. Next, in the cytoplasm, this T4 is converted to T3. And when this T3, its name 3-iodothyronine, which are uses binding, second type, binding proteins to enter to the nucleus. Okay, I just repeat this. First time in blood, T4 bind with this carrier protein, then dissociate it with this T4 and carrier protein. T4 enter inside the cytoplasm. Inside the cytoplasm, it's convert with this T3 tetraiodotyrosine with 3-iodotyrosine. 3-iodotyrosine are use this binding protein enters into the nucleus. Yes. In the cytoplasm, T4 is converted into T3 or 3-iodotyrosine, which uses binding proteins to enter the nucleus. The hormone receptor complex binds after, you see, then T3 enter inside, but inside this nucleus you bind with this receptor protein. Okay, as this number 4 is receptor protein. Okay, the hormone receptor complex binds to DNA, stimulating by synthesis of new mRNA. If you know this protein synthesis, you know, but in the cytoplasm, in the nucleus, I have this transcription and then translation. Transcription when are formation this mRNA, yes, and it's when this hormone receptor complex binds to DNA, it's stimulating the synthesis of new mRNA, new matrix mRNA, and newly formed mRNA codes for the synthesis of new proteins which are produced the hormonal effect in the target cells. As is all steps, 6 or 7 steps, I repeat. First, this T4 in this blood are bind with this plasma protein, carrier protein of thyroglobulin. Before enter in cytoplasm, it dissociated, and T4 enter inside the cytoplasm, convert with the T3, bind with this binding protein. Then after also dissociated before enter in this nucleus with T3 and this binding protein. Inside this nucleus, when enter this T3, it's found this receptor protein, which are one of this protein, which has this receptor protein, which are bind with this DNA. And from this DNA, it's from complementary principles, it's synthesize this matrix mRNA. mRNA then go from this pore of this nucleus into this cytoplasm. In cytoplasm, in the rough endoplasmic reticulum, which has this ribosomes, and in this with your 2 subunits, large and small subunits between mRNA, and synthesis this protein synthesis from translation is this process named transcription. Is processed in the cytoplasm translation. After translation are new protein synthesis, and which are thyroid hormones are a response. Okay, as these all steps or 7 steps, which are mechanism of thyroid hormones actions. Different actions you see, yes? These thyroid hormones, steroid hormones, this insulin, epinephrine and norepinephrine. Okay, understand these actions of different types of hormones? Yes or no?
Yes.
Okay, yes. If yes, it's very good. I continue about these receptors, which are different types, epinephrine and insulin.
To induce cellular responses, nearly all body cells express receptors for epinephrine and norepinephrine, called adrenergic receptors. There are 2 families of adrenergic receptors, alpha and beta, with each member of the family designated by a number. All adrenergic receptors bind both epinephrine and norepinephrine, although with different affinities. All adrenergic receptors activate G proteins. Beta receptors are coupled to adenylate cyclase by stimulatory G proteins that increase cyclic AMP activity. Alpha-2 receptors are coupled to adenylate cyclase by inhibitory G proteins that decrease cyclic AMP activity, often blocking the increases induced by other agents. Alpha-1 receptors act via the IP3-DAG and calcium second messenger system. Some cellular responses to these catecholamines are listed beneath the appropriate receptors. blood flow is redirected to working muscles where increased cardiac output delivers increased fuel and oxygen. Fuel molecules are released from stores and made available for use by working muscles. The airways dilate to maximize gas exchange. Note that all of these
No, these epinephrine and norepinephrine receptors, are two types, two family, alpha and beta. Alpha has two subtypes, alpha 1, alpha 2, beta 1, beta 2, which are, when binds this one same neurotransmitter hormones with these different types of receptors, it has different actions in the cells, and also different actions of these hormones, and also different effects. Example, when this epinephrine or norepinephrine bind with these alpha receptors, as these actions I explained, but when this phospholipase 3 activates, and which are catalyze this reaction from diacylglycerol and this IP3, when release calcium ions, and these calcium ions are sometimes calcium you won't but it's need when contraction of this muscle, smooth muscle layer which are in vessels, vasoconstrict vessels of skin, mucus, and abdominal muscles. Yes, this calcium calmodulin complex. Next, this when bind with these alpha 2 receptors also are this dissociate with this G protein, cycling convert this with alpha and beta gamma subunits. Alpha activate cycling AMP, cycling this adenylate cyclists, which are converts the cycling AMP ATP to this SAMP. SAMP after inhibits insulin secretion of beta cells of pancreas. One of these functions when this epinephrine binds with alpha 2 is inhibit secretion of beta cells of pancreas, inhibit insulin secretion of beta cells of pancreas. Yes, from negative feedback. Insulin function, is decrease this concentration of glucose in this blood. Yes. Next, this beta 1 and beta 2 receptors. Now, these receptors are location in these blood vessels, and these beta receptors, one is location in the heart, and this which is air action also are G proteins, second messenger systems, which are need this cycling AMP, and then no guanosine diphosphate, which are released and binds this guanosine diphosphate, and heart rate, force of contraction also. Heart rate increase and force of contraction also increase from this norepinephrine, are this postganglionic sympathetic neuron release epinephrine and norepinephrine, yes, which are increase heart rate and increase contractility of this. It's two effects. When increase heart rate, it's named positive chronotropic effect. Yes, when increase contraction of this heart muscles, cardiac muscles, is named positive ionotropic effect. Yes, but why? But postganglionic sympathetic neuron, when entering the heart, it regulates SA nodes, AV nodes, and also cardiac muscles. Yes, but its contraction force, positive ionotropic effect, is a sympathetic neurotransmitters. And last, these beta 2 receptors are vasodilate vessels to the muscles and dilate airways, blood vessels. Okay. Yes, understand. Yes, this about alpha.
Support the fight or flight response.
Fight or flight responses are
insulin acts primarily on liver, adipose tissue, skeletal muscle, and cardiac muscle by binding to tyrosine kinase receptors. Each tissue responds differently, and each has multiple responses. Some of the cellular responses to insulin are listed beneath the receptors. Glucose uptake increases in all three major tissue types. Insulin stimulates glycogen synthesis in muscle and liver cells, and triglyceride synthesis in adipose and liver cells. Amino acid uptake and protein synthesis are stimulated in muscle. Note that these effects support the overall goal of promoting storage of fuel molecules.
Okay, no, about this, insulin and other hormones, I explain in the next lecture. Okay. Okay, have you questions? No, ma'am. Yeah, understand. Okay, very good. I'm happy if you understand. Thank you for listening and watching me. Okay, very good. Okay, and I'm sending. I have some of the students' email, and I upload this presentation about this endocrine systems. And you prepare this first and second lecture of this my presentation, which are this about chemical structure, about receptors, and about different types of action of different types of chemical neuro hormones. Okay. Yes. Okay. Welcome. Upload. Okay. Write your email. Some of the students' email. Please write me on chat. Okay, and I send at this moment this new presentation. Okay. This email I have. Yesterday I also sending this. Okay. Okay, today I finish, my dear students. I send this presentation after 5 or 10 minutes, and see you next week. Okay. If you have questions, write me on my messenger or on my. Uh, if you want, I write my phone number and from Viber. If you want, please contact with me. I write my phone number 551 15 48. Lali Modebadze. Okay. Bye bye. Nachwam this.